This is episode 56 of the Immunology Podcast, Epitopes in the Immune System with Dr. Alessandro Sette. Hey everyone, this is Dr. Jason Goldsmith and Dr. Brenda Rout. Welcome back to the Immunology Podcast, where we have conversations with immunologists. The Immunology Podcast is brought to you by Stem Cell Technologies, a global biotechnology company supporting life science research and foster communication and collaboration in science. If you enjoy the Immunology Podcast, rate us and leave us a review. We're always looking for feedback on how the podcast can be improved and for suggestions on guests. Today, we have Dr. Alessandro Sette from the La Jolla Institute for Allergy and Immunology on the podcast to talk about his research studying the specific epitopes that the immune system recognizes in cancer, autoimmunity, allergy, and infectious diseases. We've also got our usual roundup of highlights and immunology news coming up. But first,
a reminder that the 2023 IUIS Congress is taking place in beautiful Cape Town in South Africa from November 27 to December 2nd. Early bird registration is open until August 31st, and we do hope to see you there.
All right. Well, summer is upon us. Conference season is over.
Sun season is over. We haven't had rain for like a full month in here.
Yeah, we have the same problem.
So summer finally has arrived to the Northern Hemisphere.
I want it to be at least a little cool. I'm going camping next week, but it's a LARP camp. So it's like in armor and everything called Drachenfest US. It comes from a German LARP called Drachenfest. And me and the family are going. And so, a nice, 23 Celsius would be better than 25 Celsius. Under 80 degrees Fahrenheit would be wonderful. A little bit of cooling would be good because I'm going to be running around in a gambeson and armor and I don't want to, sweat to death.
Can you please repeat what kind of camp is it? I don't think.
The LARP, live action role-playing camp. So there's 5 different armies that are all going to fight each other with foam weapons, but real armor over the course of a week. And our children are coming too. And so you have to dress up your tent to look medieval, and there's fire pits and stuff to cook with and, lutes instead of guitars.
Wow. America is a place for everything.
No, this comes from Germany. This one is based off the German Drachenfest, which is one of the largest LARPs in the world with 50,000 people at it.
Oh, wow. How kind I've never heard of it. Clearly not in the same. We're not moving in the same circles here.
Clearly
a shame.
Not. We need
but that must be something fun to at least to watch or to participate. And I do like that you're making it a family experience.
Oh,
That's kind of cool.
I mean, have you ever built in school catapults or trebuchets or any of those things in physics class where you have to launch something?
Not really. We're not taught warfare equipment in our.
Just imagine now your children can use their trebuchet building skills in real life to launch foam cows at your enemy.
You can also I don't know, maybe catapult corpses, infested with pest.
Yeah. That's a thing.
You
get
that.
That's a fake dummy corpse and you put it on the catapult and you chuck it. Yeah.
Anyway, let's move on and let's go down to our topic of the day, immunology, every day, all day.
All day, every day is immunology day. All right. I can start first. I have microbiome immunology and alpha-4 beta-7 integrin signaling as my theme of the day for both papers. But we'll start, but it's also cancer immunology. So I'm playing in all the fields here. So the first one is called, it's in cancer immunology. It's distant anti-metastatic effects of enterotropic colon cancer derived alpha-4 beta-7 CD8 T cells. So first authors are Virginia Filiu and Carlos Gomez Roca. It's in the lab of Christelle Duvod. I think there's a co-PI as well. I'm just going based off the asterisks here of Maha Ayub. And this is a fascinating paper. So it essentially, so take a step back real quick. Alpha-4 beta-7 is the integrin that is for mucosal homing, particularly in the intestine. And so what they show is that if you take, so colon cancer has really bad liver mets. And so you can do models where you inject the metastases in the liver in mice because you can't get metastatic colon cancer in mice very well because the tumors grow too big before they get metastatic and occlude the colon of a mouse and kill them. In humans, this doesn't a problem because we're bigger. And so let's say a tumor has to be 3 centimeters to generate metastases. Well, in a human, that's small relative to the size of the lumen of your intestine. In a mouse, it's big and kills them. So it's a problem of like size, not that the models aren't great. The models are actually very good, except for the size limit. So typically you'll take the cells and inject them already metastatic. And so they derive those lines and you put a primary tumor in the colon. So they show that these hepatic tumors, and they also are will kill a mouse from the metastatic disease. But if you put a syngeneic colon tumor, some of the mice show regression of the disease. So, and in regression, you see shrinking of both tumors. And they show that then if they inject this tumor in other parts of the body, not the colon, it doesn't work. But if they put the liver mets in other places like the skin, you get the protection again. The skin metastasis or extraintestinal site shrinks, not all the time, but often. And that goes along with the colon cancer shrinking. And what they found is that there's a colon-based immune system effect that is derived from CD8 T cells that are alpha 4 beta 7 positive. And if you blockade this with an anti-integrin antibody or to kill the cells, or if you use a different line that isn't the same, it doesn't work. And they do some T cell receptor mapping and antigen mapping and find that there's clonal expansion of neoantigen marking TCRs and MHCs based on the presence of this tumor, right? So based on tumor antigen in the colon, and that these immune cells are going to the metastasis and then presumably killing it. They then also demonstrate that in people who have a response tend to have more of these markers, alpha 4 beta 7 T cells in their metastatic lesions that if they're going to end up responding later. So on biopsy, they could look and measure, and then they see the response later on. And they also show that there's better response to immunotherapy in those people. And so if you break out to microsatellite stable, which is, we know it works in microsatellite unstable, microsatellite stable colon cancer, this works better. The checkpoint blockade works better if you have these cells in your tumor already from the colon-derived or colon-generated T cells. So fundamentally, a colon immune response to your cancer is predictive and therapeutic against your metastases, and that's what they then try to drive. So that is my first paper on integrin-related cancer immunology of the colon, but there's more to come, because why do it just one paper?
You gotta stay with the topic. But then, so what's also, I guess, important is how to study these models, how difficult it is to find the right models to study these diseases if you have mouse as a model organism. All right. I'm going to talk also a little bit about cancer immunotherapy. And the paper, first paper I want to talk about is called Tumor Immune Rejection Triggered by Activation of Alpha-2 Adrenergic Receptors. And this is a paper was published in Nature. First author Jing-Xing Xu from the lab of Benoit van der Einde at the Ludwig Institute for Cancer Research in Belgium, in Brussels. And this is, I think, an example of trying to repurpose drugs that are already used for other indications, and kind of initial observations, indicate that they might be useful for some other treatment. And then you try to kind of see if you can apply them more rationally and study the mechanism by which these observations occur and whether you can really take advantage of it. And so adrenergic receptors belong to the class, to the G protein couple receptor class. And they are the targets of very well-known hormones such as adrenaline and its counterpart noradrenaline. And the blockers of these receptors are used a lot in the clinic for many effects. And I think you as an MD probably know better. And the study kind of is inspired by the observation that inhibition of beta 2 adrenergic receptors have shown some kind of benefit when combined with checkpoint inhibition when it comes to cancer treatment. And so there's this idea that because these might act through inhibiting the one of the main parts of the downstream signaling of these adrenergic receptors, which is the production of the second messenger cAMP by adenyl cyclase, and by inhibiting these, in this case, the beta 2 AR receptors, you can reduce the production of this cyclic IMP. But then the alpha 2 AR receptors do the opposite, and they're kind of activated by complementary pathways. And when they get activated, they do the opposite thing, and they inhibit adenyl cyclase, and that's decreasing cyclic AMP levels. So what the researchers here thought is, well, if we know that by inhibiting the opposite enzyme, we get this beneficial effect, what happens if we actually activate the alpha 2 adrenergic receptors? And there's a bunch of molecules that do this. And so they use a mouse model, or they use a bunch of tumor models in mice to look closer into this option. So basically, they have a lot of mouse models, a lot of different tumor cell lines. They also work with a xenograft model in which they inject cell lines, so tumor lines derived from patients into mice, and then they also do immunodeficient mice, and then they introduce PBMCs from donors to initiate an allogeneic kind of T-cell response against these tumors, because now these donors and the tumors are not matched. And they use that as models to kind of see whether by adding these alpha 2 AR agonists, they can modify the anti-tumor response. And interestingly, they basically see that for a wide selection of tumor models, both cell lines and patient-derived xenografts, adding alpha 2 AR agonists actually seems to reduce tumor growth and increase survival of the mice. And they have this huge figure with lots of different survival curves and tumor growth curves showing it for several different models. And this also seems to be beneficial in the case of certain tumor models that are known to be quite resistant to immunotherapy, which seems very interesting. Some of examples of this agonists are molecules such as guanabens, clonidine, and guanfacine. I guess that's a way of pronouncing it. And so they, throughout their study, they look into different of these molecules. Some molecules they not always act the same way, but they all really seem to suggest that activating this pathway is beneficial for the immune response. So, and on top of that, they have a mouse that is knockout for this alpha 2 adrenergic receptor, and they see that basically the effect of this agonist is basically abolished. So you really do need this enzyme to be present, not on the tumor itself. Doesn't matter. What matters is that it's present in the immune system of the recipient. And when they start looking into the immune response in these tumors, one of the things they notice is that they have an increased infiltration of CD8 cells and a higher activation of both CD8 and CD4 cells. And this kind of and this, I guess makes sense if you start seeing a better rejection of the tumors. they show they use an MC38 ova model, so an MC38 cell line expressing ova that is partially responsive to anti-PD-1 therapy. MC38 is known for being very quite aggressive, and they show again that by using, in this case, they focus on clonidine as one of the inhibitors, that really increases the survival of the mice. But then when they look into the immune cells inside the tumor, one of the things they notice that they see better survival of CD8 cells within the tumor, and a reduction in the number of immunosuppressive myeloid cells. And this is very critical, because when they look into digest material from the tumors, they do single cell RNA sequencing, and they kind of characterize how the tumor responds to treatment. It's really clear that in at least in this settings, macrophages represent a large majority of immune cells that are infiltrating the tumor. So about 87% of CD45 positive cells. So, and interestingly, they see that one of the major changes that they see in this, in the immune cells of treated tumors, of tumors from treated mice, is a huge, an important shift in the phenotype of this macrophage, of these infiltrating macrophages, towards a more phagocytic, more pro-inflammatory profile. And these, they suggest, seems to be the reason that kind of the part of the way this change in the T cell response is driven by having a better immune stimulation from macrophages. Or we could also say less immune suppression, because also macrophages and other myeloid cells in the tumors are famously known for often providing very strong immunosuppression to the tumoral, inside the tumor. They also look into some signatures from tumor from human tumor data. I have to say that I found that less convincing, but it is really hard to pick up these signals when in this big databases. So I don't take that so much against them. So what I also thought was a little bit not ideal is the fact that they give really large doses of these agonists as at the kind of the rate of 5 milligrams per kilogram per day in IP injections. And that actually, interestingly, seems to, well, maybe not interestingly, but expectedly, that actually has also effects on the mice because they become kind of what's the word, sedated, because that's what these agonists do. So that's also not super easy to treat the mice with this agonists. And apparently even these alpha-2 agonists increase the aggressiveness of male mice. So they were, yeah, Jason, you don't see, but Jason is agreeing. So apparently they could only use female mice for these experiments.
Well, what's interesting is that clonidine's dose in people is, a lot less. A milligram of clonidine is a lot of clonidine.
Yeah.
I dose it, 0.1, 0.2, 0.3 mgs in a human in a day.
Yeah.
So you said 1 mg per kg?
5 mg per kg.
I don't even believe that they're activating their specific receptor then.
I mean, that's a fair, I think that's a fair criticism. I have to say that after I read the paper, that my first impression was like, oh, this is very straightforward. But then I'm looking a little bit into the details. I also thought, whoa, that's a lot. And they do mention it in the
but why not use physiological doses? That's weird.
For lower doses, they don't see these effects. So that's why. That is the quick answer to your question.
Something is a
yeah. So would we treat people with these agonists at these concentrations, given that it will also have kind of severe, not severe, but clear effects on this sympathetic, system? Like probably going to have effects on the CNS. I don't know. You know better, but it was still an interesting read, and they tried a lot of different models. So, I mean, A for effort.
Yeah. Well, that's very interesting that the dosing, well, I don't know, GPCR signaling guy here, the dosing bothers me emotionally just a little bit. But what really I do emotionally like is more alpha 4 beta 7 signaling.
No,
Oh,
no more,
hey, it's
please.
course
No.
anti alpha 4 beta 7 antibodies course through my blood at all times. So I'm particularly invested in it. So
Okay.
this one is a microbiota modulated checkpoint directs immunosuppressive intestinal T cells into cancer. It's in cancer immunotherapy. The author list is Maureen Fideli et al. Corresponding author is Lawrence Zitvogel. And this was a really interesting paper for me because it's the opposite story of what we just heard. So there's this known effect that they explore in more detail that antibiotics treatment before immune checkpoint therapy can have worse outcomes to it. So that's an interesting fact. And so they're trying to go, hey, what's causing that, right? What's going on here? So given that whole scenario, what they found was that the other side of alpha 4 beta 7 in epithelial cell is MADCAM1, which is the component on the epithelium, right? Alpha 4 beta 7 integrin being on the immune cells. So they find that normally speaking, you have eubiosis with antibiotics, or if you throw a specific bug in on top of things, you get more of this genus called enterocluster. And that presence of that downregulates MADCAM1. Downregulation of MADCAM1 allows IL-17 positive, ROR gamma T positive, alpha 4 beta 7 positive Tregs. So alpha 4 beta 7 Treg, but thinks IL-17 and ROR gamma T to leave the gut, and they go to the tumor. And in the tumor, they are immunosuppressive. So normal immunotherapy can be effective, and they do this with lungs cancers, right? So they do these with tumors in systems in mice that are orthotopics. This isn't colon cancer tumor. This is just colon immune cells. But if you give antibiotics or an alpha 4 beta 7 antibody to block it, or anti-1 MADCAM, or do genetic ablation, these immune cells do not home to the gut. They go to the peripheral tissues where they're immunosuppressive and ablate the effect of immune checkpoint therapy, anti-PD-1 therapy. And they have been able to see that in people given antibiotics, outcomes are based partially antibiotics, but then predictively even better based on if they see this dysbiosis of this organism showing up. They can feed mice this organism. It causes the effect as well, right? You don't have to just do antibiotics. This grows up in the absence. When you give antibiotics to get dysbiosis, and this overgrows. Or they can see that in people, more of this bug existing is a negative prognostic indicator. So in the end, you have a bad acting microbe that will downregulate MADCAM1 after antibiotic usage, which causes immune cells to leave the intestine and go to the periphery tissues where they are immunosuppressive. They do a lot of work. They show there's no difference in clonal expansion or expansion generally of the T cell compartments based on this bug. It's all about homing. And this screws up the homing, and things leave the gut that shouldn't and become immunosuppressive elsewhere. So where in the gut immunosuppression, good. In cancer, bad. All based on the antibiotics you took for that infection you got with your cancer, or before your cancer, the week before.
Wow. What a story. I like it.
Yeah, I thought it was pretty. It's the opposite side, right? It's the other paper was these alpha 4 beta 7 activating immune cells in the colon had effects elsewhere. And in this one, it's these suppressive ones leaving that are bad.
Yeah, and isn't there the option of having like kind of similar to your previous story, having they don't see any effector T cells doing the same and helping the anti-tumor response? Is that a thing at all? I mean, it makes sense that you have a lot of FOXP3 positive suppressive regulatory T
Right.
cells. These are the ROR gamma T FOXP3 positive,
Right.
double
Right. And
positive.
they're in the gut normally. That's where they should
Yeah,
be. But if you
yeah,
have them leave because you get rid of MADCAM1 because your antibiotics dysbiosis, or if you just force it, or if you have
yes.
alpha 4 beta 7 flowing through your veins, like some of us, anti alpha 4, then they can go to where there's tumors and then they make it so your checkpoint blockade therapy doesn't work.
But aren't they any effector cells that live in the gut that do the same, that have the same happens to them and then they move?
Yeah, but those effector cells, because this isn't about colon cancer, unlike the first paper where in colon cancer, those effector cells are good actors elsewhere, right, on
Mm-hmm.
the tumor. This is like lung cancer transplant models or people with lung cancer generally. And so there's
Mm-hmm.
no prime
Antigen specificity.
activating antigen specificity in the T cell compartment of the gut that's leaving, but there is just generic suppressive capacity leaving.
Yeah.
Because while there is antigen specific suppression in the gut, there's a lot of generic suppression just because there's so many antigens. You can never have them all. And so those leave too. And now you have suppression everywhere.
Wow. I mean, that's so important, right? It's obviously a very active field at the moment, understanding like what is the microbiome that you want to have if you have cancer, especially if you're undergoing certain treatments and immunotherapies. I am just really excited about us understanding this and, in the future, getting some kind of microbiome treatment before you start your checkpoint inhibition for your tumor or something like that. And then that has shown to improve your chances of survival. So, yeah, again, microbiome one, cancer. Well, in this case, microbiome zero, cancer one, because they played against us. But
But the
I'm
other
sure
one
we,
might not. Yeah, it's always an interesting story, right? Microbiome immuno-oncology is a very hot field. And I can say
yeah.
no more.
All right, nice. So, you know what's another hot field that I like is
T
cell
cells.
therapy, T cell therapy, gene therapy, all those interesting new novel treatments. Okay, so I'm going to talk about it's a short paper. It's more of a report of a clinical trial of the first patients from a clinical trial. But I think they made quite a splash a couple of weeks ago when the initial, press release was released. And I was following the story and I thought it was very interesting. And now finally we have the official paper on the New England Journal of Medicine. And the article is called Base Edited CAR7 T cells for relapsed T cell acute lymphoblastic leukemia. First author Robert Chiesa, and from last author is Wasim Quasim and they are from the Great Ormond Street Hospital for Children at the NHS in the UK. And I like the story because I think it's, I mean, there's many clinical trials going on when it comes to cell therapy, but every once in a while, there's one that really, is something new, something they put together a lot of novelty. And in this case, they had some really nice results for some of the patients. So I thought it was worth discussing. So this is a phase 1 clinical trial showing the effect, initial results from using an allogeneic CAR T cell treatment for T cell leukemia. Until now, all of the approved CAR T cell therapies are, targeting mostly CD19 and other antigens that are present on B cells, and they're useful for these B cell leukemias. But T cell leukemias are always complicated also because CAR T cells are T cells. So if you look for something that is expressed in T cells, you need to find a way to prevent the CAR T cells from killing each other. So that's what they do in this paper, in this study. They use CRISPR, but not traditional CRISPR that generates double-strand breaks, but using cytidine deamination guided by CRISPR, these famous base editors, to mediate more precise editings that do not include a double-strand break in the genome of the cells, and this has improves the safety profile of these products, because then you don't have the chance of having weird chromosomal translocations, which are known to be an issue with traditional CRISPR approaches. And they make you can often obtain very precise editings, like one base to another, in which you generate a stop codon, or you mess up with an intronic, like a intron exon border, preventing proper splicing. And this, now they show in this study, they show that you can use these tools to generate a highly potent T cell product that can be used against T cell lymphomas. So what are these they call CAR7 T cells? Basically PBMC derived T cells. These are from non-related donors. So this is an allogeneic product in which they use base editing to modify bases in the genes of the TCR beta chain, to remove the endogenous TCR of the cells, to prevent graft versus host disease. They also, of course, target CD7, which is the target of the CAR the cells will express, so to prevent fratricide between the CAR T cells. And they also deplete CD52 to give them some resistance to treatment with alemtuzumab, which is used to deplete the leukemic cells as well. Basically, they show, using this deaminase, they use a deaminase enzyme, and they generate a cytidine to thymine conversion, therefore messing up codon here and there. And they show that they can very efficiently generate these cells. On top of that, they transduced a CAR against CD7, and they end up with these CAR7 T cells. They show that they can do it. They show that they prevent chromosomal translocations by using the system, that the cells look really good. And they can then, in the end, after they expand the cells, they can remove any cells that still have this because they're all external antigens. They can remove any cell that still has a TCR or still has CD7, and then they can get a product with a very high purity with what they want. So they treated three young patients with this therapy. And I think the first patient is the one that made the most when it was in the news, and this patient was very well studied, because she responded extremely well to the therapy, and so basically she was treated with a round of this product, which again is allogeneic, so it's like from a third-party donor, and she recovered very well. And what was interesting, so of course there were some levels of some toxicities related to the therapy, but everything was very manageable. And this girl really went into to remission. And then after, I think it was 28 days, so they did a treatment of 28 days in which they assumed no residual disease. And then, because this is important, they treated the patient again with lymphodepleting chemotherapy. They remove the cells, and then they do another round of bone marrow transplantation. And this is important because otherwise you need to recover your T cells eventually, and that's how they do it. Otherwise, you would be the problem with this particular CAR is that it basically can target any T cell, and then you would be forever lymphodepleted in that sense. But they show that after they do a second round of lymphodepletion, they get rid of the therapy, and then they put again bone marrow transplantation, and the patient recovered very well. This was not the case for the second patient, however, and this I think was good to see how risks are taken. The second patient could not survive the lymphodepletion, the initial lymphodepletion, and the extended lack of T cells, and contracted a very severe Aspergillus infection in the lungs. And unfortunately, I don't remember if it was he died of complications from not having T cells. So it's very hard. These patients also were in risk for CMV, reactivations, EVV activations, all kinds of these opportunistic infections. When you have a depleted immune system, they can be very dangerous. And there's a third patient that also was treated with the therapy, but has been recently treated. So he did. After 28 days of treatment, they do show that they couldn't detect any leftover disease, and now they are going further with the new stem cell transplantation to see if they can recover the patient's immune system. So I think I really liked it because I like cell therapy, but I like it because it's an allogeneic therapy. That means that you don't need to get the patient's cells, which can be very limiting. Unsurprisingly, PBMCs from oncological patients are often not the best starting material for these therapies. You can make these therapies and you can have them stored, and then you can give them to many patients from one batch. You can treat many patients. And the fact that you can kind of do the treatment until there's no residual disease, and then pursue a new lymphodepletion and bone marrow transplantation, I think is really, really good. A really good way of managing the inescapable toxicity of T cell directed immunotherapy. So I'm looking forward to the rest there. They want to treat 10 patients in total for the phase 1 trials.
Yeah, that's super interesting. I think you make a point about the risks, but it sounds like there could be a really new, cool technology here. It's very exciting to hear. Just being able to target T cells, double-edged sword, but their unique use of genetic engineering, I think, is very exciting. All right. Well, we're going to be speaking to Dr. Alessandro Sette at the La Jolla Institute for Allergy and Immunology in just a moment. But before we get to that, if you're enjoying the conversations on this podcast, we invite you to read more interviews on the Stem Cell Technologies webpage. In the Immunology Profile series, immunologists tell their stories, discuss their research, and voice their thoughts on current topics in immunology. You can find these interviews at stemcell.com/immunoprofiles.
Joining us today is Dr. Alessandro Sette. He's professor and head of the Division of Vaccine Discovery at La Jolla Institute of Allergy and Immunology in California. And he's going to be talking to us about his research, but also I would like to hear more about his career in general. I think he has a very interesting history, and also a lot of this research is close to my heart in the sense of understanding how T cells see the world. Professor Sette, thank you so much for joining the Immunology Podcast.
Thank you, Brenda and Jason. It's great to be here.
So you are originally from Rome, so you're from Italy, and you started your career in immunology at a very exciting time. And I know that some of your early work understanding how MHC2 presents antigen to T cells, and I think it's very interesting. And it happened at a moment in which we really didn't understand much of what was going on. So I was wondering if you would be maybe to share with us a little bit about your experience in those early days immunology in the mid 1980s, and also how you pivoted through to industry. So you worked also when you moved to the United States. You ended up working for many years at a company developing immune therapies or understanding the immune system for therapy. And now you find yourself working at La Jolla Institute. So maybe if you wouldn't mind to share with us a little bit your career arc, because I think it's very interesting. Our listeners would really like to know.
Thanks for asking. So I went to school in Rome, in Italy, and I graduated, and then I left to go to Denver at the National Jewish Hospital, and I had a postdoc with Howard Gray, that was at that point chairman of the Department of Medicine at National Jewish Hospital. Very exciting times. Was Kapler and Marak were there, and with Howard Gray and a number of very prominent investigators. So I was very, very shocked. I arrived in the middle of January in Denver, never seen so much snow on the ground. I never saw people that actually wore real cowboy hats. I thought I was in a movie. I loved it. And it was very pivotal time, as you were mentioning. At that point in time, believe it or not, it was not clear that the function of MHC was to bind peptides. And Howard lab and Emil Yunanu's lab, which recently passed, really generated the biochemical evidence that that was the case. And it was very controversial because other people thought that no, that there were two different receptors, one for MHC and one for peptides. And I remember we presented our data. I was terrified. I was a young postdoc, the World Meeting of Immunology, just right after one of the main proponents of the fact that it was not true. And but it was one thing that was, I remember in particular, was very criticized because we were doing receptor-ligand interaction with scattered plots. And I could never show that 100% of the MHC that we had in our test was active, was capable of binding peptides. Of course, now we know that it's because the MHC is fully saturated with endogenous peptides. But we could show that maybe 5% of the MHC was active. And so people were criticizing this and saying, oh, this is not MHC binding of peptides, it's probably some contaminant. And I remember I redid those gels, I don't know how many times, and I was telling Howard, I can't make it any more pure. I mean, this is what it is. It's not. So anyway, those were exciting times. And as you're saying, then this was in 1985 to 88. And in 88, became clear that maybe this could be used, this understanding could be used to maybe have some applications. And I was very interested in that. And Howard Gray started this company that was funded from money from San Diego. And so we moved in 88, we moved to La Jolla and we started this company called Cytel, which was very interesting. And then Howard left and went to the La Jolla Institute, and I stayed on. And then eventually I was interested in vaccines, and we spun off with two other colleagues, and we founded a new company called Epimune. So I was at Epimune until 2003, and I was a chief scientific officer there. So I really loved to do clinical trials and testing things for real in humans. And at the same time, though, the company became more and more focused on manufacturing and clinical trials and investors. And I'm kind of a technology guy, I'm a research guy. And so I at the end of the day, I said, well, this is a very important job to take this further, but it's not me. So I told them, look, bye. And we left in very good terms. I mean, because obviously I wanted them to be successful. And I went back to academia, and I've been there since 2003 at the La Jolla Institute. But still, in a way, a lot of what I brought back some of my biotech mindset into academia. A lot of what we do is actually also still really focused on having clear goals for project timelines, program managers, and doing a lot of contract work, because also, if you're doing immunological evaluations for a vaccine, you need to get it done on time so that the information can be used for the next step. It's not up to you to say, well, I was something else that was more interesting today. No, we got to get it done.
So I was wondering if you could, I'm also in biotech. So I was wondering if you could talk a little bit about applying that biotech mindset to academia and how it's led to some of your more recent successes.
Well, I mean, that's a good question. I guess in some of the work we did in COVID in collaboration with Shane Crotty, really maybe one thing that was influenced by a biotech mindset is the fact that you identify what needs to be done and you put together the expertise that is necessary. So one of the reasons why we were able to move fast is that a number of different things came together, from collaborators at UCSD that had access to samples from people that were infected and then later on vaccinated, to Shane Crotty's lab that had this tremendous expertise on B cells, the interaction between antibody and T cell responses. And my group that had the expertise of characterizing T cell responses and making peptides, identifying epitopes. And so basically, you just, rather than trying to learn yourself is more fun, right? Because you can put the postdoc there, and then after 6 months, they'll figure it out, and it's a good learning experience. But if you want to get it done, you assemble a team of people that are the best in the world to do what needs to be done, and they can do it fast and move. And then also you have very clear timelines for when step A is going to be done, so that people that need to do step B will be able to take it on.
Maybe this is a good time to talk about a little about that research that you did around COVID and the implications of finding not only T cell immunity against COVID, but also some level of cross-reactivity between other coronaviruses, with immunity against other coronaviruses and SARS-CoV-2. I remember that I read that the paper that you published, I think that was the one with Shane Crotty, in which you showed that there were many epitopes that were shared from common cold coronaviruses, and then the people already were reactive against these epitopes, naive people, the people that hadn't been exposed to coronavirus. So maybe it's a good time to ask you about, what do you think? How do you think that research came about, and what do you think, now that we have a lot more studies understanding T cell responses against coronaviruses, where are the main learnings that we got from our study of T cell responses against COVID?
Right. Yeah. So that observation came about really early on, and it was the negative control, as sometimes happens in science, at the unexpected. So we were measuring these responses from people that had been infected with COVID, and so we need a negative control. And so we thought, well, as a negative control, we'll get samples that we had banked from years ago before, so they could not have possibly been exposed to COVID. And that was going to be the baseline negative control. And we did those experiments over and over and over again, because some of those people had actually perfectly good memory responses that recognized some of the SARS-CoV-2 peptide sequences. And so that was the basic fundamental observation of this cross-reactive immunity. And this was quickly also, other labs were coming up with the same observations, Thiel's lab in Germany and many others, and had essentially the same thing. And so this was, in a way, a pivotal observation, but then came back, I think, with full significance of that was appreciated later on. I mean, if I can basically outline I think where we at with that, there has been now a lot of different studies and data that suggest that T cells can have a fundamental role in modulating disease severity. In some cases, may have a role in actually preventing infection, but prevention of infection is mostly mediated by neutralizing antibodies. And what was seen early on after the vaccinations started, that the neutralizing antibodies waned more rapidly, and the T cells actually were fairly constant. And in fact, that was what's seen also in the field, so to speak, where it was clear that even before variants, in a few months, the protection against infection was decreasing, but the protection against disease, severe disease, was maintained. And so that's kind of the role of T cells in a nutshell. And in fact, this was further correlated by the variants, because the variants escaped neutralizing antibodies and infections went through the roof, but the variants do not escape very much T cell responses, and protection was maintained. So at that point, then you can go back on this observation, the different, that there is this pre-existing memory, and it was mapped to, but this pre-existing memory, at least in some cases, clearly mapped to other coronaviruses, common cold coronaviruses, other beta cervicovirus, beta coronaviruses, and so forth. And so the concept is that not only the T cells can deal with variants, but maybe there is cross-reactivity of T cells across different coronaviruses, which is interesting. And that is actually one of the main interests of the lab right now, is this concept. So if there are regions that are highly conserved across all the different coronaviruses, maybe we can focus the immune response on these conserved regions. And the value of that is that would be a pan-coronavirus vaccine component. And I want to be absolutely clear, but I'm not advocating this should be done instead of antibodies. I love antibodies. Antibodies are good. If you have neutralizing antibodies or even non-neutralizing, it's probably very good. We should do both. But at the level of T cells, this is very interesting, because there is a lot of concern about other, specifically, for example, other cervicoviruses that may be lurking in bats or in other animals. So that this could be really a way to cover your bases against any other coronavirus or cervicovirus that could be jumping out and causing a new pandemic. And so that is, I think, a very important component. And again, this vaccine component would not necessarily prevent infection, but if it does prevent disease, that's good. In a way, it is a situation a little bit where we are now, right? Where people still get infected, but the death rates, for example, in the US, at the lowest level ever. So it's still clearly lots to be done in COVID, long COVID, huge problems. So I'm not saying everything is solved and now we should rejoice, but to have that component, a T cell component, or a number of different mechanisms that can deal with disease severity, regardless of infection, is key.
So one of your key ways you get all these discoveries is your epitope work. I was wondering if you could describe, for people who aren't familiar with how you actually do that type of hard work, can you describe it and the technology a little bit and how you're able to, as you said, understand that there's specific repeat epitopes in coronaviruses that are shared or what have you? Because I think that key structure function, almost biochemistry, molecular biology, those techniques and tools are hard, and that's kind of one of the things you're known for. So how do you do it? How do you do the magic?
Right. It's from our point of view, one of the beauties is actually it's not that complicated. In a sense, it is complicated, but the process is fairly linear. So you need sequences, of course, because if you don't know what the sequence of a virus is, that's the end of the story. But if you know the sequence or the sequences of a family of viruses, so let's start from one sequence, right? So Wuhan. So you had that sequence, and then we have algorithms that scan the viral sequences and determine what are reasonable candidates for being recognized by human T cells. We factor in HLA because, as you know, of course, T cells are HLA restricted, and HLA is highly variable in humans. So we look at the HLA space and incorporate this in our predictions. And then we make those peptides, and we make a lot of them. And then we put those peptides, we offer those peptides for recognition from people that have been infected or are convalescent, and ask, what does the memory T cell see? And so that's your screen, so to speak. And so one key component there is that this can be done fast, in the sense the sequence, again, in the case of SARS, the sequence was available rather quickly, and we can make peptides very quickly. And you can do work in the lab without having to deal with the virus. I mean, some of these nasty pandemic viruses they have all sort of issues, BSL-3 or 4 or whatever, and so safety issues. We don't need to deal with any of that because we can just make short 9, 10, 15 amino acid fragments, which are completely, obviously safe or not in any way related to a live pathogen in terms of biosafety considerations. And so we can, and that was the issue again with SARS-CoV-2. We can get going very, very quickly. The other component is we look at memory T cells, and key is the word memory. So we can look, the good thing of memory T cells is that they stick around. So we can look after the virus is gone, and the memory T cell is still there, and tell us what is being recognized. And the last point, which is also very interesting, is you can look at different outcomes. So you can say, okay, what does the T cell response look like in people that are going to have or have had a more severe outcome versus people that did well for SARS? But this is a general concept. So, for example, you can say, what does a T cell response looks in someone that has severe asthma in response to pollen, as opposed to people that don't have severe asthma in response to pollen? So you can ask, what is the difference at the T cell level between disease and health, or bad disease and not so bad disease? You can ask, in the case of dengue, what's the difference between the people that have hemorrhagic disease versus people that get infected and do not have that degree of ravaging disease that requires hospitalization. You can do that in Parkinson. What's the difference between people that have Parkinson and people that don't have in terms of their capacity to recognize fragments from alpha-synuclein or whatever? And then hopefully your vaccine looks like the good, not the bad, right? You're trying to recreate a good immune response, a response that resembles what has occurred in people that successfully fought the disease.
Yeah, and I think that's a theme that we see a lot when it comes to vaccination, but also therapeutics using monoclonal antibodies. We have had guests that look for those people that are convalescent that survived the infection, and they try to find antibodies that are recognized and they're naturally occurring, and they're recognizing that particular pathogen. But it is always very hard, right? Because we still don't completely understand. Most of this understanding, the recognition or these interactions between a TCR and an antigen requires still a lot of experimental data, right? We still need to do experiments, and this can take. The amount of different TCR sequences is enormous. That a repertoire of a person is very, very large. Even if you have a person is convalescent, or it takes a lot of work to try to find those TCRs or trying to understand those interactions. And I think this is, a lot of computation is required to maybe really mine for the hard to find specificities, or oftentimes it's not obvious. Also in the case of maybe tumoral responses, in which you have a lot of these T cells there and you don't know what they're recognizing necessarily. I just bring this up because as part of trying to advance this field and trying to understand epitopes and how they're recognized, you also have as part of your contribution to the field, we are part of the group that is behind this database that is called the immune-epitope database, and I've used sometimes. So it was very, very interesting to see that the face behind the tool. And I don't know if you maybe want to talk a little bit about your motivation for, or why do you think it's important to have these databases, and whether you think they are fostering collaboration, or in case maybe some of our listeners would be interested in using such a database, what do you think it's important about having these tools out there?
Right. Yes. So the IDB, as you mentioned, is immune-epitope database. It has also been up from early 2000, and it's freely available to everybody, and has all the epitopes from infectious disease, allergens, autoimmunity. And now there's a sister database called CEDAR for the cancer component. And it's a catalog of all the information that is published in the literature. So it's not our data, it's everybody's data. And the motivation for that, it stands back again from our point of view from the early 2000s. Already, actually, when I was at Epimmune, it was very hard to keep things straight because we did so many experiments with different epitopes, and then other people also had experiments with epitopes. And so we'll say, well, when was this tested? What's the data? Is there any binding data? Is there any T-cell recognition data? And so it became clear that to really have an ongoing effort would be extremely beneficial to have a database where you could actually query and extract that. And so then when I went back to academia, at the same point, very coincidentally, the NIH had an interest in developing this database, and so we did. And the fundamental concept of this database is really, it's an epitope database, but it's not. It's really an assay database. And let me explain. So at that point, when we were starting to say, okay, let's make a database of all the different epitopes, essentially two ways to do it. You could have some group of very wise men and women meeting once a year under a tree and going over and decide what is good data and what is bad data, and these are the good epitopes, this is the bad epitopes. And I thought that was not the approach. And so what we said, we're just going to curate everything that the people say, and then empower the user to make their decision. And so basically, if you're interested in, I don't know, epitopes against derived from influenza, well, okay, maybe you don't believe any experiment that does not use live virus. Okay, you can search for that. Or we only want to see things that are in humans but not in mice, or only things that measure interferon gamma and so forth, so that the database has all the actual assays, and you can actually search for the assay that fit your taste. And I'd like to do the example in a way is like if you do a search of PubMed and say you do a search on PubMed of tyrosine kinase beta, whatever. You just want to know everything that is known about that. You don't want to know which papers the PubMed staff thought were pretty good. And it's your call. You get all the results, and then you figure out which one you like, which one you don't like, and you may narrow or broaden the search as you please.
So I think we could talk a lot longer about epitopes, but we do unfortunately get to have a time limit for these conversations. So at the end, we like to ask a question that's not quite as science related about our guest. And so the question for you is, when you're not mapping the world's epitopes, what do you do as a hobby, or wish you had more time to do? Because epitope mapping takes a ton of work, as we just discussed.
Yeah, I mean, so one good thing maybe about COVID is actually it increased our capacity to work remotely. And so actually it has been easier, not just for me, I mean, in general, for people to work and actually work more sometimes, because wherever I am, but also to have more freedom. I love doing lots of different things. I'm still have a lot of connection with Italy. I still run a farm in southern Italy where I go there every once in a while. It's a interesting dimension, very non-epitope related, but I love it.
What are the crops of your farm, if I may ask?
There's wheat, pigs, some olives, some almonds. Yeah. And the farm has been there forever. And, the guy that runs the farm for me, his grandfather ran the farm for my grandfather. So it's a very special place. That's one thing. And then the other thing that I do, I love fencing. I always fenced, and I still compete in fencing tournaments with dubious success as I'm getting older, but I still do. Next month, I'm going to be actually at the national championship for Epi in Phoenix, and that, to a degree, also created a relationship with the San Diego Opera. So, as you know, there is in a lot of operas, there are some swordmanship, some fencing, some fights, and so forth. And so we develop a relationship with them, and often we go and do operas. We don't sing. I don't sing. I could not sing for the life of me. But we do supernumerary roles. So last couple of months ago, I did the Tosca. I was a custodian in Castel Sant'Angelo, scrubbing the floor of the blood after the executions. And my wife was particularly pleased because she said, "Oh, I pay good money to see you on your knees scrubbing the floor, because certainly you don't do that at home."
Oh no, that's so funny. That's awesome.
For the pigs, is it Iberico ham or any of that?
No, Iberic. That's from the Iberic Peninsula.
Spain, right?
Oh my gosh,
that's my
Jason,
bad. No, what was it?
Save it.
What
Save it.
Italian pig?
Yeah, but the prosciutto. Usually the prosciutto, the good prosciutto is up north in Parma and so forth. No, these are good pigs.
I'm more interested. I'm more curious about the olives and the almonds. I mean, of course, world famous Italian olive oil.
Do you give extra virgin olive oil as a gift to people who graduate, like a little bottle to take with you?
No, actually, sadly, I don't bring it. It's complicated, and you have to have
export laws and all that.
Yeah. And I just buy it at the grocery store like anybody else.
So, well,
terrible. You have your own farm and you can't bring it over. I know the rules of international commerce, right?
But you do get to enjoy it when you go visit. So, so I guess that's good, because then you keep it. It's something to look forward to.
Yeah. We're going now in July.
Wonderful. Oh man, if only. It was such a pleasure talking to you today and hearing about your research and your story as a researcher. And thanks for sharing with us today. And Jason, anything you want to add?
Oh no, that's it. I will have to go review my ham geography, but I'm just going to blame it on being Jewish.
Please, yes, no. There are Jewish people all over the world, so that's just
no. We don't eat pork that much.
Yeah,
Sure. My knowledge of ham is low. I'm just going to blame it on that and run away.
Sure. But we have chicken. Yeah.
That's kosher, right?
Yeah, you're good. All right.
All right. Go ahead, Jason and Brenda, thank you very much for having me today. I really enjoyed it.
It's a pleasure.
A pleasure.
This brings us to the end of our show. Don't forget to subscribe to our newsletter at www.immunologypodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at @immunopodcast or via email at info@immunologypodcast.com with feedback or to suggest guests. See you next time.